Good morning, good afternoon, and good evening, everyone, and welcome to the 13th webinar in the Cardiovascular Connections 2022 series. Today's webinar is titled Cardio-Renal-Lymphatic Disease, featuring Dr. Giacomo Rossitto, Assistant Professor at University of Padua. Today, Giacomo will share his research on heart failure and the related lymphatic dysfunction in patients. Before we get started, we would just like to acknowledge our partners at the American Physiological Society and the European Council for Cardiovascular Research, and also to thank the sponsors of this webinar series for making it all possible. And now I'm pleased to bring Giacomo on to join me. Hi, please begin whenever you're ready.
Right. Hi, everyone. I'm absolutely excited of this opportunity. And I will try my best today to share with you some ideas on why I believe that many of the cardio-renal and vascular diseases that we tend to approach are also lymphatic in nature, and that's my task. So, as Sydney mentioned, I'm a clinician. I work in Padua as a consultant, and I also have a honorary research position at the University of Glasgow, where I previously had my PhD training. And I'm particularly happy of this opportunity as a shared webinar between the American Physiological Society and the European Council for Cardiovascular Research, also thanks to Insight Scientific, to really contribute to these cardiovascular connections, which I truly believe are absolutely key in the progress of our science and what makes our clinical practice incredibly exciting nowadays. So I really have not many conflicts of interest to share. This is my agenda. I will try my best today to discuss with you these few points, and I would just like to start from what is probably sort of background to convince you of why lymphatics are important in what I call here cardio-renal vascular disease. It's increasingly true for us clinicians that the kidneys play a key role in the pathophysiology, the physiology and pathophysiology of cardiovascular disease in general. And well, I would say that physiologists and people from your societies were really aware of that long ago, certainly before clinicians start to make things real in everyday's practice, but I will touch upon that again later on. So this is what Guyton proposed long ago as a sort of scheme to depict the very complicated cardiovascular physiology. And you may see that it's not just a matter of heart function or vascular function, it's a matter of many different things. One of these, of course, include the kidney dynamics right on top of the cartoon. And I was lucky enough during my training to get in touch and get training from great experts in some bits of all these complicated pictures, like Professor Rossi in Padua or Professor Dallas in Glasgow. And really had the opportunity and luck to meet others that substantially contributed to the understanding of one or many of these tiny boxes. And particularly in relation to what I highlighted as tissue fluids, pressures, and gels, Jens Titze, Professor Titze, now in Singapore, is really someone who made a tremendous contribution over the last couple of decades in the field. So he's the one that approximately 20 years ago made some seminal observation that he further developed way more. And he first noticed that whenever you were running some sort of metabolic experiments, and those experiments were lasting long enough, the salt loading of animals and humans really at some point was no longer paired by accumulation of water as the accumulation of sodium happened in tissues. to a point that he and his group suggested at that time the existence of a sodium reservoir in our body with the ability to store significant amounts of sodium that he proposed to be osmotically inactive, so not paralleled by similar amount of water. And I'm really making a very, very long story short, but he managed to first propose that the largest site for that accumulation was the skin. So he not only proposed and identified preclinically all the theoretical and experimental background to this concept, but really made it possible to see that tissue sodium for us. That was something that was really changing the physiological paradigms for many of us. And by using a technology based on magnetic resonance spectroscopy, he could show that 23 sodium signal was much, much higher in the skin, but not just skin, also in the skeletal muscle, as you can see here, of an aged and hypertensive patient compared to a young, healthy control. And that was first shown in approximately 10 years ago in, as I said, aging and hypertension, particularly in secondary forms of hypertension or refractory forms of hypertension in terms of treatment. But then there was accumulating evidence showing pretty much the same in many other settings, including CKD, including diabetes, including heart failure, including systemic sclerosis, multiple sclerosis, lipedema, lymphedema, and so on. And many of these studies gave us also the opportunity to see that interventions could reduce this amount of tissue sodium in the different tissues, skin and muscle. So what we did was to take this concept and just go a little bit back to the drawing board, and that's something that I will try to do with you today. So please bear with me. I will be very basic at the beginning, but that's really in the interest of understanding how we approach this issue. And you may see here a general piece of tissue containing some extracellular component that, as we all know, is pretty rich in sodium, and an intracellular component that is rich in potassium, and that is really traditional physiology, what we all know. So the two components are pretty much one the mirror of the other, but if you sum up sodium and potassium, you reach a fairly similar value that is in between 140 and 150 millimoles. So if you take a piece of this tissue and you really make a smoothie out of this, you get some sort of orange solution that is darker or lighter depending on the relative proportion of the yellow and the red components in this solution. But the sum of the two, of sodium and potassium, and this is physiology, still would remain pretty much around 140 to 150 millimoles per liter. So if you just do the same, rather than pretending to make a smoothie of a tissue or chemically digesting a tissue, you do it with a box cell, as you would do in the 23 sodium MRI. That's exactly the same. So you're basically identifying a volume of tissue, and within that volume, if that tissue is particularly high in cellular mass, you will have a pretty faint signal for sodium. If the volume is poorer in cellular mass, or vice versa, is richer in the extracellular volume, or some moiety of extracellular volume was added on top of that, of the physiological tissue, then your signal for sodium concentration will be higher. And that is true also if you visualize it as radiologists would do, and is really happening regardless of hypertonicity or isotonicity of that sodium accumulation, and really depends on the compartmental distribution of your tissues. So what we did was to do a similar sort of analysis by direct chemical analysis rather than voxeling tissues. We took tissues and digested them into nitric acid and measured the exact water content of those tissues and sodium and potassium content in the very same pieces that we collected from rats at the beginning, as shown here. And we used spontaneously hypertensive, stroke-prone rats compared to WKY controls. And we salt loaded both groups to see the effect in terms of sodium content and water content in tissues on all these different organs. And I would really like to thank people from my previous group, so Guto Montesano, Adam Harvey, and Sean Merry from Glasgow, who did fantastic job during these experiments. So what we found was that indeed the sodium content was increased by salt loading in the salt-sensitive animals. And that was true for skin, for liver, for lung, not really for myocardium, unless we aged the animals to a point of having them almost 1 year old. But that was not independent of water. That was really paralleled by water, as you can see in the middle row. And the sum of sodium and potassium really stayed constant across groups, regardless of the strain or the loading protocol, thus suggesting isotonic phenomena happening, not really anything that was water independent. These data really matched the findings we had in humans. As a clinician, my target, of course, is translating evidence to humans, and we managed to collect skin biopsies from the arms of hypertensive patients. And once again, you can see that the distribution of the sodium plus potassium sum was really within the values consistent with isotonic physiology. And the increase in both sodium and water was correlated with the increase in age, and that is pretty much in line with the physiological change in the compartmentalization of tissues with aging. And of course, in a young subject, you would expect much higher cellularity than you would expect in an older subject. And that's why we observed that increase in sodium. And these data, of course, correlated with surrogates for total body sodium, like NT-proBNP, or parameters related to vascular function, like pulse pressure here. So interestingly enough, if you go deep into many of those 23 sodium MRI studies that highlighted this incredibly high prevalence of excess tissue sodium in many different cardio-renal vascular diseases, to a point that tissue sodium correlated with left ventricular hypertrophy in CKD patients much stronger than blood pressure or other usually explored clinical correlates would do. If you just substituted the tissue sodium for tissue water in your prediction model, the result was exactly the same. Or in other patient population, like patients with heart failure, studies performed with punch biopsies, very similar to what we did in our hypertensive patients. The amount of water, total water concentration in the tissue, and the amount of glycosaminoglycans, which were supposed to be the site for water-independent binding of the positively charged sodium, were actually very, very closely correlated. So probably that binding was not limited to sodium, but also to the high amount of water present in the tissue. So my conclusions from our experimental findings were that high tissue sodium concentration does not necessarily equal hypertonic, so water-independent concentration, and therefore, given the high prevalence of this phenomenon observed with 23 sodium MRI in many, many patients that we see on an everyday basis, that tissue sodium excess, at least in part, reflects changes in tissue architecture and or edema. And in most of those patients, that's not a degree of edema that we can pick by normal physical examination, for example, by pushing our thumb on someone's leg. It's something that in most of the cases was totally subclinical, so we were totally blind to that amount of expansion in the intracellular volume. And we concluded that human hypertensive skin made no exception to that. I would like to highlight that possibly skeletal muscle does, and that's something that has been really nicely shown in the last months by work from Professor Week. He did a chemical analysis very similar to those that we did, and he, I would say, that came to fairly and overall similar experimental conclusions. As you may see here, there is no real difference in the sum of sodium and potassium across the experimental groups that he used, namely low sodium diet, high sodium diet, and DOCA salt. And that was true in skin, skeletal muscle, and cardiac muscle. But they did one additional extremely clever approach. So they treated these animals with tracers in order to track the extracellular and intracellular volume fraction in tissues, and they sliced the tissue in order to get chemical analysis, which were not just informing on the whole piece of skin or muscle, but were really targeting that specific layer. And they found out that the increase in sodium was not only in the interstitial fluid, where actually not much hypertonicity was detected anyway, but really also within the cells in the intracellular space. Although, of course, an expansion in the extracellular volume was identified too. So I think that the two phenomena are not mutually exclusive. Probably they complement each other, and they just open up the door for additional research in the topic to understand what that excess sodium is doing in the extracellular space, and also what that is doing in the intracellular space, depending on the cellularity of tissues, which of course are different. When you compare a piece of muscle to a piece of skin, the amount of possible interstitial expansion is certainly different, and therefore mechanisms there for handling that excess sodium can be different. But overall, I believe that we do have reasonable evidence to believe that that excess tissue sodium is reflective of edema, and as I told you, that is extremely prevalent in cardio-renal-vascular disease. I believe that you would not be surprised in seeing that if that is the case, and if 23-sodium MRI have shown excess signal in diabetes, in hypertension, and in patients with non-end-stage chronic kidney disease or end-stage chronic kidney disease, if all these conditions at some point will evolve in a syndromic condition that is called heart failure, it's probably not surprising that most guidelines-based and guidelines-supported treatments for these diseases are mostly natriuretic in nature. And that includes RAS blockers, mineralocorticoid receptor antagonists, glyphosins, and of course, traditional diuretics. So basically, by targeting these axes, which are common across these conditions, whether they are at the stage of risk factors or at the stage of overt disease, certainly that is making a difference in robust outcomes, as shown by a number of clinical studies now. So if that is the case, if edema in a clinically overt or subclinical form is so important in cardio-renal-vascular disease, then it's probably worth going back to the pathophysiology of that. And that is what brings me to Starling Landis equilibrium. So this is the real mechanism of microvascular exchange that I was taught when I was a student And one first point that I would love to make during this webinar and to share with you is that this is no more the case. So Starling and Landis did fantastic studies, which are really seminal and really unsurpassed, and devised this concept by which at the site of the capillary beds, a good proportion of fluids would get out of the capillaries into the interstitium from the arteriolar side of the bed, and a similar proportion of fluid would go back in and reabsorbed into the venular side of the microvasculature, based on the equilibrium between osmotic and hydraulic forces. So fact is that once actual measurements rather than presumptions were made on those parameters, so the oncotic pressure and the hydraulic pressure, and I will keep it simple, but that also includes the role for the glycocalyx and how that modulates the diffusion of proteins across the capillary bed. We came to realize that that bimodal distribution of fluid filtration and reabsorption in the capillary bed is really not true. So in most conditions, with important exceptions, but like hemorrhage, for example, or some specific vascular beds like in the kidney, but in most other conditions, the filtration is happening throughout the entire microvasculature. And there's no substantial reabsorption at the site of venules, but there are other key vessels that really play a key role in draining that constantly filtrating fluid. And these vessels are lymphatic vessels. So these are not ancillary structures, but are really key structures in physiology, taking care of all that amount of fluid that is constantly filtered throughout all our organs. And just to give you a quantitative picture of that, for you to believe me, that means that that lymphatic contribution to the refill of the intravascular space is really what we call a plasma refill rate in a sense. And that, in order to really appreciate the quantitative relevance of this, I would show you the data from these very old studies performed in humans, where people cannulated the thoracic duct, and they realized that even in control patients or compensated heart failure patients, the amount of drainage of fluid through that thoracic duct during the day was in the order of 60 cc's per hour, which is really in keeping with a normal urine output for a normal subject. And if the subject was in a condition of decompensated heart failure, that value increased much, much more. And all this is possible because lymphatics are incredibly sophisticated structures, as you can see in this video. So I came to learn that lymphatics are capable of a systolic activity, of a diastolic activity, of pacemaking in order to drive these mechanics. They are made up of single functional units which are in series, and they're called lymphangiones. And all these lymphangiones are separated by unidirectional valves, like the one that you can see in the video. And these valves are maintained in a healthy state via a number of genes and signals, including the flow through those vessels, as reviewed in great reviews very, very recently and depicted here. So these vessels are so sophisticated that you can really draw a pressure volume curve really identical to what our colleagues in the cardiac cath lab would do with myocardia. And work from Mike Davis' lab, and he's a scientist that really got a lifetime achievement award from the lymphatic community for his tremendous contribution to this understanding of lymphatic mechanics. he showed that these vessels are really responding to preload and afterload, similar to what a myocardium would do. These are extremely sophisticated structures. And this, of course, opened the concept of lymphatic function rather than simply lymphatic anatomy, as reviewed here by Joshua Scanlon, who comes from Mike Davis' lab. And in this review, he highlighted how much this function includes elements of complexity, which goes from the spaces that these terminal lymphatics open in order to absorb the fluid from the interstitium, the generation of systole, which starts with a dedicated pacemaking in these vessels, the coordination of this pacemaking activity and the coordination of contraction, of course, the competency of these valves and the permeability of vessels. And other great scientists from Mike Davis' lab have specifically contributed to these aspects, highlighting the channels that are involved in the pacemaker generation of the depolarization wave, and the role of valves and their competence in the conditions of health and disease, like Scott Xavier and Jorge Castorena Gonzalez. So all this sophistication is why I at some point decided to buy the idea put forward by Professor Stan Roxon from Stanford, who suggested that, in a sense, each edema can be regarded as a lymphedema. And so if you just consider any closed compartment like this square that depicts a piece of tissue, you will have some sort of input, the arterial, and of output, the lymphatics. And if you really think of lymphedema as clinicians would do, so just only when something like Elmin's, or most commonly surgeons, are damaging the lymphatic drainage, that is probably like taking the Brownwald textbook of cardiac medicine and just read the physiology chapter and the cardiac tamponade chapter. There's much, much more in between those two extremes. And basically, whenever even a healthy lymphatic network cannot match an excessive input, or an unhealthy lymphatic network cannot match an even normal arterial input, then edema develops. And that is why, in the context of diseases where edema and subclinical edema are so prevalent, that's why lymphatics are important. And in order to convince you of that, I will start back again with Jens Titze, who very nicely showed approximately 10 years ago that in the skin that accumulated the amount of sodium that I alluded to, there were very specific lymphangiogenic mechanisms, including VEGF-C signaling and VEGF receptor 2 and particularly 3 receptors in these vessels, that whenever blocked, led to a phenotype that was characterized by excess tissue sodium accumulation, and in particular salt-sensitive increase in blood pressure values. And I would like again to mention that probably the ball is still in the air. These are not conclusive data because, once again, from the very same investigators who used different animal models of dermal lymphatics excess or defect, they couldn't replicate the same changes in blood pressure or the same changes in lymphangiogenesis. But it is certainly not a simple topic to run conclusions on, because of course hypertension is really a multifaceted disease with many different players involved. And there is evidence from the renal side of the story from Brett Mitchell and Joe Rukowski, Texas A&M, who managed to work with animals with renal selective overexpression of vascular endothelial growth factor D, which is another specific lymphangiogenic factor acting through VEGF receptor 3. And this renal-specific overexpression not only prevented the development of hypertension in their animal models, but also reverted and improved the pressure control in animals that were already made hypertensive. And at the beginning, most of the evidence really focused on the immune side of the story of what we all know is absolutely important in the development of target organ damage in hypertension. But most recent evidence clearly pointed also to a totally different sodium handling from those renal lymphatics expanded kidneys. And there is a lot of data and evidence pertaining renal lymphatics that I don't have the time to summarize here today, but I would really like to point you to these fantastic reviews produced by different groups really around the world. What I would just like to mention today is that what we generally use in clinical practice for getting some relief from edema, so loop diuretics like furosemide, really appears to act in a way that we were not expecting. And this work by Shelton and Kohn, published in the American Journal of Physiology, Renal Physiology in 2020, really highlighted how the lymphatics depend on NKCC activation and all the downstream signaling in order to generate pacemaking and in order to generate valuable contraction. And I can tell you that when those data came out, I was really not surprised, because I understand this is not robust evidence. The N is just equal to 1. But when I was training in Mike Davis' lab, we really had to go with some bumetanide, which is similar to the furosemide. And we really had very similar decreases in the amplitude of contraction on vessels, which were not renal, but were peripheral. So that is something that in the future we will possibly take into consideration when treating our patients. So most of those data, of course, are from preclinical models. And as I alluded, my contribution to the fields really wants to be the translation of that to my patients. So what we did was a study in the extreme of a continuum spectrum, which starts from diabetes, CKD, hypertension, but ends up with heart failure. And we focused on a specific group of heart failure patients, which are those with preserved ejection fraction. And these patients are well known to be patients affected by microvascular disorder in terms of function and anatomy of rarefaction of these vessels. But the perspective has always been on the arterial side of the story. So we managed to compare these patients with age-matched and sex-matched controls, and to compare them in terms of plethysmography for microvascular function and skin biopsies. So we adapted a very specific protocol in order to explore the microvascular exchange, and I will explain to you what we did by basically applying increasingly high pressures, which were still in the order of the venous pressures, to the limbs of these patients. We got these bumps that you can see on the left, and each bump corresponds to the distension of veins, which were getting some opposed drainage by our cuffs. But on top of these bumps, we also observed at the different cuff pressures some further increase in the tissue volume that was not related to the intravascular distension of these vessels, but to the interstitial accumulation of fluid. So by plotting the slopes of those interstitial components, what we found was something very similar to Landis graphs, basically depicting the constant of filtration and how the interstitial accumulation occurred as a function of the pressure in our cuffs. And this point of intersect, which is particularly important, that is higher of the venous pressure, was the isovolumetric pressure. So the point when the lymphatic drainage could not cope any longer with the amount of fluid that was filtering through the vessels. Contrary to what we were initially expecting, so some excess filtration in our patients, shown in red here, compared to the controls in black, there was not really a steeper constant of filtration in those patients. So there was not excess microvascular extravasation of fluid, but certainly the isovolumetric pressure was much smaller in patients compared to control, suggesting that their breaking point, when the lymphatics were no more capable of draining the extravasating fluids, occurred at much lower pressures. And this was supported by evidence of not only blood vascular rarefaction in the skin, but also lymphatic rarefaction in the skin, particularly for vessels with small diameter, and by gene expression data, showing here that genes like PROX1 and LIVE1, which are respectively one of the key drivers for lymphatic development and one of the most used markers of small lymphatics, were substantially reduced in our population compared to controls. So we came up with the idea that despite an even smaller amount of microfiltration due to the vascular rarefaction of the arteriolar vessels that these patients experience, the drainage offered by lymphatics was even more impaired in these patients, so that the lymphatic reserve in heart failure appeared to be reduced. And surprisingly, well, I would say interestingly, probably not surprisingly, this is a concept that others are exploring in terms of treatment. And there is now the opportunity to directly target interstitial rather than intravascular congestion in these patients by means of tools that are generating reduced hydraulic pressure at the site where the thoracic duct drains into the traditional classic vascular system. And with this tool applied to ships, the investigators could show that the amount of extravascular lung water was reduced by the activation of this device that was basically bypassing the and leading to a facilitated drainage of fluid from tissues and to reduce the extracellular water. This is of course experimental, but we're really looking forward to see the human translation of this. So for those who are particularly keen to look at the heart itself and not only the periphery, which is certainly a meaningful and really important approach, I would point you to this great review that has just been published by Kathleen Caron, and that summarizes the work over the last few years done by many groups, and particularly those by herself, by Eva Brackenhill and Guillermo Oliver. They really contributed incredibly to our understanding of cardiac lymphatics that are specific in terms of their own mechanics, and they appear to be extremely important not only in heart failure, but also in conditions of ischemic heart disease and myocardial infarction. This is true also in terms of atherosclerosis. We're now aware that lymphatics are involved also in the trafficking of lipids, not only from the gut, but also from atherosclerotic plaques in vessels. And the field of lymphatics is really expanding in many fields and in many organs that were not considered part of this story until a few years ago. And these are just a couple of more reviews that I would like to suggest to you for consideration. So in relation to my presentation today, and I really hope that I managed to raise some interest and to convince you of the relevance of these vessels in the pathophysiology of cardiorenal vascular disease. We know that excess tissue sodium is prevalent. We know that, at least in part, that excess tissue sodium really represents edema, either clinical or preclinical, and if that is the case, what is really handling that edema is the role of lymphatics. And lymphatics are incredibly beautiful structures, sophisticated structures. I believe they are really wonderful. sophisticated to a point that we know they're important. We still need to better understand to what extent they are important and possibly impaired in their intrinsic function in cardio-renal disease, in order possibly to tackle that function and get some further improvement in the treatment of our patients in the future. And with this, I would really like to thank the people that trained me and convinced me that all this field is extremely exciting and worth investigations that I'm sure many of you will possibly in the future contribute to. And I would like to thank you all for the attention, and I'm happy to take any question that comes from the audience. Thank you very much.
Hi, Giacomo. That was a fantastic talk. Thank you so much. Yeah, let's jump right into our Q&A session. Okay. The presentation mostly covered tissue fluid homeostasis, but lymphatics are known to also modulate inflammatory cells in the tissues. So can you comment on that, please?
Oh, this is a great question. Absolutely relevant. We all know how much inflammation plays a role in the pathogenesis of cardio-renal disease in general, and organ damage and so on. I deliberately touched only upon the hydraulic side of the story. Of course, part of that modulation of the interstitial fluids and cells is regulated by lymphatic function. We now start to know that diseases which are really inflammatory in nature, like Crohn's disease, for example, are impacted by lymphatic function. That's something that we will learn in the next few years for cardiovascular disease too.
Fantastic, great. All right, our next question is from Jeffrey. Can you provide details on the skin samples, anatomic location, gender, BMI, etc.? Did you track angiogenic and anti-angiogenic genes in the skin samples of soluble plasma?
So thank you, Jeffrey. Well, of course, all those data are published. So I would really point you to that paper that was published in JACC really pretty recently. The anatomic location for the biopsies that we took on patients were on the arm, so sites were not traditionally supposed to be that involved in the clinical edema that we would consider as clinicians, which is much more commonly in the lower limb, for example. And those patients tended to be in decongested conditions. So those are baseline features that we observed. Gender and BMI, patients were pretty balanced and matched for that, as I alluded. And we did track the angiogenic and anti-angiogenic axis by tissue gene expression, and in the hypertensive patients also by measuring soluble VEGF-C in serum, not plasma, in our samples.
Okay, that's great. Thank you. We have another one from Jeffrey. Is eNOS active in lymphatic endothelial cells?
eNOS is indeed active and is one of the key modulator of lymphatic relaxation and also valve function. I would really point you to a paper that was published in PNAS in 2016, and also to the comment that Professor Davis made on that paper. That is a hugely important component of lymphatic function that is dysregulated, for example, in diabetic lymphatic, as shown by Josh Scallan, for example, and is one of the master regulators of the complexity that I showed you.
Okay, makes sense. All right, we have a long one here. Jill asked, is lymphedema lymphangiogenesis, an indicator of lymphatic system reacting to being overwhelmed? So does high VEGF-C indicate increased lymph flow or efforts to address increased interstitial fluid, whether successful or not in increasing lymphatic fluid transport capability in response to edema? Let me know if I need to repeat any part of that.
Yeah, thanks, Jill. That's a great question. So that's probably the very traditional chicken and egg question that we see in human research. So it's not something that I think we have dissected entirely. What I personally believe is that, of course, VEGF indicates a reaction, and I believe that reaction is what our body always tends to do in order to maintain homeostasis. There are points when we fail at that. So even the renin-angiotensin-aldosterone system activation is physiologically good, but at some points becomes bad. We know that VEGF-C through VEGF receptor 3 also modulates function, not only lymphangiogenesis. We don't know how to exactly correlate peripheral values or even tissue values with the flow at the moment, particularly in pathophysiology and not only in physiology, at least to my knowledge.
Okay, thank you. We have another one here from George. How much of the reduction in lymphatic capacity is due to downstream venous hypertension, i.e. right atrial pressure and compliance?
That's another great one. I cannot give any quantitative answer to that. I personally believe that when I showed you the modulation of lymphatic function depending on the afterload, that is of course relevant whenever you run those myography experiments and you increase the output pressure to those vessels. Of course, contraction is made more difficult. So part of that story pertains excess afterload. The adaptation to that afterload in the long term and the maladaptive response to that is something that we are studying at the moment.
Okay, that's great. Thanks. All right, we have another a little bit longer one. Jay asks, there are many studies on the total interstitial fluid volume, extracellular, but do we know the volume of the lymphatic vessel system, including lymphangions to the largest ducts, example, thoracic and right lymphatic?
There are some estimates. There are some reported numbers which approximate 8 liters of fluid handling by the lymphatics per day. I try to be more conservative, and I showed you what I truly believe could come from a human experimental setup when they cannulated that thoracic duct. Those volumes were those really coming from the main collector, where pretty much all the lymph is basically drained into. And I showed you that it is at least up to 1.5 liters per day in normal subjects, but can go up to almost 500 mLs per hour when your system is overloaded. So I think that this is close to what probably the lymphatic vessel is the lymphatic system is handling in our body.
Okay, that makes sense. Thank you. Anna has asked you, is there anything patients can do to increase lymphatic drainage of excess fluid?
Well, this is another great question. So how to move it forward and go to therapy? So I just alluded to one investigational approach, which is at the moment under investigation, as I said, and that is by facilitating the drainage of the lymphatic system via the lymphatic duct. If you just move your perspective to the more traditionally called lymphatic patients, so those with the lymphedema, for example, post-surgical lymphedema, we all know that the therapy is largely supportive in nature. There are, of course, some attempts to restore the patency of severed vessels by surgery. There are attempts to increase the lymphangiogenesis at times when those vessels are damaged. There are different results in the literature. One recent study didn't show much improvement with VEGF-C therapy coupled to surgery, but the study was small and with large heterogeneity in the groups, so conclusions may be biased by that. So at the moment, the increase in lymphatic drainage is mostly achieved by mechanical tools, so massage and compression garments and those kind of approaches. But there are other tools that are under development, and I'm really positive that in the next few years this will change the field.
That's amazing. Yeah, we are getting lots of great questions. So I'll just let everybody know, continue to submit them in, and Giacomo is doing a great job at responding. So our next one from Raphael: is there a nervous regulation of lymphatics? Pardon me.
Thank you, Raphael. There is indeed. We have variable evidence on the different peptidergic or non-peptidergic signaling that is active in lymphatic vessels. There is a great review by Peter Russell published in Cardiovascular Research last year, I think. Indeed, the adrenergic system regulates the lymphatic flow. We don't know exactly how our treatments that impact on the adrenergic tone, like beta blockers or possibly even alpha blockers in the clinical practice, really affect that clinically. But I really happened to have a chat with a colleague who deals with sympathetic denervation in patients, and we really wondered what the impact would be on lymphatic flow. We don't know at the moment.
Okay, thank you for that. Our next one up here, Emilio says, since beta blockers are commonly used in heart failure, have you explored the consequences of this blockade or the alpha adrenoceptor blockade in the lymphatics?
Yeah, thank you, Emilio. I think this is really similar to the previous one. There is a bunch of experimental evidence, mostly preclinical evidence, I would say, as Peter Russell reviewed in that paper I alluded to. We don't know exactly what happens in the patients that we treat.
Okay, thank you. Ned has asked, how might you image or measure preclinical edema?
Oh, thank you very much, Ned. This is something we're still learning. I think that I will really give credit to Jens Dietze here. He did a fantastic job in making it possible by at least visualizing tissue sodium, which is not a perfect marker of preclinical edema, as I tried to explain, and part of that sodium might be stored elsewhere than the interstitium, but certainly, at least in part, as I stressed, does represent interstitial edema. So if we manage to visualize or at least quantify sodium properly in tissues, that can be a good surrogate for that. And I believe we're not too far away from that goal in our even clinical practice, I would say.
Okay, fantastic. All right, we've got another long one a little bit here. Jay has a follow-up question. You specified lymph flow rates in normal, controls, and heart failure patients, but I was wondering the total volume of the lymphatic vessels in normal and heart failure, assuming lymphangiogenesis occurs to expand the total system volume due to edema.
I would. Oh, thank you. Maybe I misunderstood the question, so I'm happy to expand my answer. I think there's a huge amount of plasticity in that system. So we really have limited information on a whichever way you want to define it, baseline condition. We know even less how much that's supposed to be baseline condition change whenever there is VEGF-C signaling, an attempt to respond to increased afterload. So we don't have clear figures for that. What we do see in, for example, in biopsies from lymphedema patients is that these vessels are distended. So probably the volume that is stored, if I'm not sure that is the right word, but let's call it this way, in the vessels really depends on the global fluid status, body sodium, for the reasons I alluded to, and it's probably really, really plastic and really, really variable across patients, ages, BMIs, possibly gender. So I would expect a lot of variability. I don't have the exact numbers, sorry.
Okay. No, I think that was great. All right. I think we can squeeze one more question in. Raphael has asked, are there portal systems in the lymphatics?
I'm not aware of that. But there's another review I would point you to that was published in JACC by Max Itkin last year, was rather discussing the lymphatics in the portal system, so in the liver. That is another organ where the lymphatics seem to play an important role, and you may find some information in that review. I'm not aware of a lymphatic portal system, if I get it exactly right as you suggest. That's what I know. Sorry.
That's okay. Maybe some avenue to explore in the future for
Sure.
someone.
There's plenty of avenues in the field.
That's right. Okay, I think I will make that our very last question. So thank you so much, Giacomo, for sharing your knowledge and expertise. It was really a pleasure to have you with us today.
Thank you very much, and thank you all. It's been a pleasure for me. I'm super happy of this shared effort by the American Physiological Society and the European Council for Cardiovascular Research and Insight Scientific, of course. So thank you all for being with me today.
Thank you so much. Thanks. All right. And a big thanks also to the audience here for participating. In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and the European Council for Cardiovascular Research. We look forward to having you with us again soon.